Good day everyone, we are Group 4 of Section N-3L. Together with my partner Brandon Felizarta, I, Jefford Monteza, will report to you the post-laboratory reports of Exercise 9 that is all about flowers. Flower is produced by an angiosperm. Sexual reproduction takes place in this structure. The vegetative shoot is analogous to it, and the individual flower sections are leaf-like in appearance. The typical flower is an assemblage of sterile and fertile plants borne in an axis, the receptacle, and supported by a stalk, the peduncle. Flowers may be solitary or clustered into an inflorescence. The fertile parts are also called as essential parts, which are mainly involved in reproduction are the stamen, collectively called as androecium, and the pistil or gynoecium. The flowers of different plant species have morphological differences. The source of variation are the number of insertion of floral parts, sex expression, floral symmetry, ovary position, and type of placentation.
The objectives of the study are the following. First is to identify the importance of plant reproductive system. Second is to know the correlation between pollinators and the morphology of the plants. Third is to illustrate the anatomical parts and its embryogenesis. And lastly is to identify the plant development. For Section 1, the floral morphology of the plants, the methodology of the digraph plant are the following. First is to examine the Hibiscus rosa sinensis. Second is to draw the figure or the flower, and lastly is to label its respective parts and sexual organs. Figure 1 shows the labels and parts of the hibiscus flower, where it shows the petal, which are the segments of the corolla of a flower. The sepal pertains to the leaf-like enclosing the petal. Calyx consists of fused petals. Epicalyx resembles the calyx, but consisting of a whorl of bracts that is exterior to the calyx. Bracteoles, which is the small bract. Corolla makes up the petals of the flower, while the perianth pertains to the collection of both calyx and corolla. Stamen forms a ring around the pistil consisting of the pollen, anther, and filament, and style pertains to the stalk structure atop the ovary, while the stigma pertains to the area where pollen germinates, and lastly the pistil as the female organ of the flower consisting of the stigma, style, and ovary. Figure 2 shows the parts of the male reproductive system of the hibiscus species and dicot plant, showing its fertile structures and its bithecal nature. Theca pertains to the pair of microsporangia, and having two thecae, as observed in the flower, means that it is bithecal or tetrasporangiate. The male reproductive system is the stamen that forms a ring around the pistil, which consists of a pollen that is located inside the two pollen sacs of the anther and responsible for germination. Anther pertains to the opening when it is ripe, releasing for pollination, and the filament, which is the stalk. The anther is the collective term for pollen and pollen sacs or microsporangia above the filament. Figure 3 shows the female reproductive system of a dicot plant, showing its fertile structures. The female reproductive system is the pistil collectively, which consists of the stigma, style, and ovary. Stigma pertains to the apical protruding structure at the center of the flower, while style pertains to the stalk-like structure below the stigma, and lastly, the ovary is beneath the style that constitutes the ovules.
The methodology for Section 1 Part B are the following. First is to examine two species of monocot plants flower, namely the orchid flower and a flower of Arachis species. Next is to draw the monocot flowers, and lastly is to label its respective parts. Figure 4 shows the photo of an orchid flower and its parts. Labellum, the inner tepal, is enlarged that often serves as landing platforms for its pollinators. The pollens in anther are aggregated into pollinium covered by the anther cap. The anthers of the androecium are fused with the gynoecium style and stigma, forming the gynostomium or the column. Figure 5 shows the parts of the flower of Arachis hypogea. Peanuts flowers, or any flowers in the legumes family, have one large banner or standard, two lateral wings, and keel, which encloses the staminal tube at the distal end. The stigma is surrounded by 8 anthers, which is composed of 4 oblong-shaped and 4 globular-shaped anthers. The ovary is located at the end of the hypanthium inside the bracts, which joins with the reproductive branch. Inside the hypanthium tube, a style is seen from the apex of the ovary extending above the anthers to form stigma. The methodology for Section 1 Part C are the following. First is to examine Oryza sativa spikelet using online photos. Next is to draw the rice spikelet, and lastly is to label its respective parts. Figure 6 shows the parts of Oryza sativa spikelet. Grass flower differ from monocot and dicot flowers in terms of parts and structures. Spikelet is a term associated to the small flowers or florets which develop into seeds or grains. Each spikelet is separated by glumes, and each floret borne on top of a stalk or a chela is in turn protected by 2 additional bracts. The lemma, which is often ond, it is the hair-like or bristle-like structure or appendage on a larger structure, and the palea. Immediately above the palea are 2 scales known as slodecules. Basically, the grass flower has 2 fused carpel and 3 stamens.
Section 2 pertains to the variations in floral structures, which is about the relative positions of the ovary. The methodology for the relative positions of the ovary for this section are the following. First is we need to examine the flower specimens. Secondly is to cut the flower longitudinally. Then to draw and label its respective parts, and lastly is to identify the relative positions of the ovary. Figure 7 shows the hypogynous nature and anatomy of a lanzonas flower in its longitudinal section. In hypogynous flowers, the perianth and stamens are attached to the receptacle below the gynoecium. The ovary is superior to these organs, and the remaining floral organs arise from below the point of origin of the carpel. While Figure 8 shows the epigynous nature and anatomy of squash species flower in its longitudinal section. In epigynous flowers, the hypanthium is fused to the gynoecium, and the free parts of the sepals, petals, and stamens appear to be attached to the top of the gynoecium. Lastly, Figure 9 shows the perigynous nature and anatomy of a peach flower in its longitudinal section. In perigynous flowers, a hypanthium, or the floral tube formed from the fusion of the stamens, petals, and sepals, is attached to the receptacle below the gynoecium and surrounds the ovary. The ovary is superior, and the free parts of the petals, sepals, and stamens are attached to the rim of the hypanthium.
Section 3 is the microsporogenesis and the microgametogenesis. These processes takes place in the male reproductive parts of the plant. The methodology for Section 3 includes the following. First is to search for online references of the 2 processes, microsporogenesis and the microgametogenesis. Next is to illustrate the distinctive features in the 2 processes. And lastly is to label its structures and parts. Microsporogenesis is a process where a microsporocyte in the anther divides by meiosis 1 and 2 to give rise to 4 haploid microspores. The cell mature into pollen grains, and after this process, it will be followed by microgametogenesis. In this process, the series of events undergone by the nucleus inside the pollen grain leading to the formation of sperm nuclei. This process has 2 rounds of mitotic cell division of the pollen nucleus into the tube nucleus and the generative nucleus. Unicellular pollen undergo the first mitosis producing bipolar pollen, then afterwards that pollen undergoes the second round of mitosis producing a tricellular pollen. After the pollen grain has landed on the stigma, the pollen tube grows downward through the style. While the pollen tube elongates, the generative nucleus divides into male gametes or sperm nuclei, while 3 nucleated body inside is the male gametophyte. These nuclei are released from the ruptured tip of the pollen tube into the embryo sac when the tube reaches the micropyle of the ovule. Section 4 is the megasporogenesis and the megagametogenesis. These processes takes place in the female reproductive parts of the plant. The methodology for Section 4 includes the following. First is to search for online references of the 2 processes, megasporogenesis and megagametogenesis. Next is to illustrate the distinctive features in the two processes, and lastly is to label its structures and parts. Under megasporogenesis process, it produces 4 megaspores from the megasporocyte in the nucleus through undergoing meiosis 1 and 2. 3 of these spores degenerate and only 1 remains functional. On the other hand, megagametogenesis gives rise to the egg or the female gamete. It undergoes 3 consecutive mitotic cell division in the functional megaspore to produce 8 nuclei and 7 cells. An egg, 2 cells of synergids, 2 polar nuclei in 1 cell, and 3 cells of antipodal. The egg and synergids are lodged on the micropylar, while the antipodals are on the chalazal end.
Section 5 pertains to the inflorescence, where the flowers are borne in groups of clusters. The methodology for the inflorescence are the following. First, you must go to the backyard and observe flowers formation. Secondly is to examine the flower specimens, then to take photographs, and lastly is to identify the type of inflorescence. Figure 12 shows the composite flower of Tridac's daisies in longitudinal section showing its carpel and stamen. Composite flowers means that it is a flower that is made up of multiple simpler flowers, or the inflorescence of the pseudanthemum type. The carpel pertains to the female reproductive organ of the flower, while the stamen pertains to the male reproductive organ, which is shown in the figure as a disc flower. Figure 13 shows the spiky inflorescence of Anthurium species. A spike inflorescence pertains to a raceme, but the flowers develop directly from the stem and are not borne in pedicels. While figure 14 shows the flat umbel inflorescence of the Bermuda grass. Flat umbel pertains to the indeterminate inflorescence, where clusters of flowers made up of several short stalks that spread from a common point resembling the ribs of an umbrella. Then figure 15 shows the corymb inflorescence of a common yarrow. Corymb flower pertains to a cluster where all the flowers are at the same level, with flower stalks of different lengths forming a flat-topped flower cluster. Next, figure 16 portrays the solitary cyme inflorescence of a gumamela flower. Solitary cyme means that the development of an inflorescence axis is a single terminal flower along the leaf axis. Lastly, figure 17 portrays the corymbus cyme inflorescence of a sampan flower. A corymb pertains to a raceme indeterminate inflorescence where the pedicels of the lower flowers are longer than those of the upper flowers, so that the inflorescence has a flat-topped appearance overall.
Section 6 talks about flowers and its pollinators. Plants cannot move from one place to another to hunt for food, nor to find their mates. With this, they must meet these requirements through their growth responses and the structures they generate. Many angiosperms have acquired a collection of characteristics that effectively allow them to move around in search of a partner. By luring insects and other animals with their blossoms and regulating their behavior such that cross-pollination or cross-fertilization occurs often, angiosperms have overcome their rooted status. They have become just as mobile as animals in this regard. Flowers and their pollinators co-evolve and to achieve this. The methodology for Section 6 includes the following. First is to collect the studied specimens. Next is to use the pollinator guide and then make it in a tabular form. And lastly is to identify its corresponding pollinators. Table 1 shows the collected flowers and its corresponding pollinators in tabular form. First is the Anthurium species, and the first pollinator of this plant species is the beetles. This is because Anthurium have large, bowl-like shaped flower and has ample pollen in it. Birds are also attracted to the Anthurium since its flower is scarlet in color. Also, it does not have any odor. Next is the Cynodon dactylon. The pollinator of this plant is solely the wind since it has dull green in color and has no odor nor nectar in it, hence not attracting any other pollinators other than the wind. The pollen are not sticky. This is the continuation of Table 1. Shown is the flower of Achillea millefolium, and its pollinators are the butterflies and bees, since it has bright white in color and has a fresh scent to it. The pollens are also limited. Since the type of inflorescence of this plant species is corymbed, the other flowers attached serve as the landing pod. Next is the Hibiscus rosa sinensis, which has a solitary cyme inflorescence type. The pollinators of this plant are the butterflies and birds. This is because these pollinators are attracted to the red color it has. Butterflies are the pollinators of Ixora coccinea, and this is because butterflies are attracted to the color of the flower, which is red. Aside from that, it has ample nectar and is hidden in the narrow tube of the flower. Other flowers can serve as the landing pod of the pollinator.
For the post-laboratory study questions,
For post-lab questions, question number 1, explain why the flower can be interpreted as a modified branch or shoot. According to Moseth of 2006, a flower has an enlarged condensed axis called thalamus. In the thalamus, it has a very short nodes and internodes. At the nodes where the flower whorls can be found, hence the flower can be interpreted as a modified shoot. Since the thalamus continues to elongate, it basically shows internodes, making a flower a modified shoot. For question number 2, what becomes the ovary, ovule, ovary wall, and integuments after fertilization? Ovary tissue, where the ovule or ovules are attached, develops into a fruit. The fruit may be dry or fleshy. It also encloses the seed, and on the other hand, ovules develop into seeds and ovary walls into a pericarp of the fruit. Moreover, the integuments become the seed coat where it protects the seed. It is according to Beck of 2010.
for question number 3, what is the fate of each of the 8 nuclei in the embryo sac? The embryo continues to germinate and undergo embryogenesis, which is a continuous process to become the seedling. The polar nuclei and egg cell or nucleus both participate in the double fertilization event. The egg cell is fertilized by one of the 2 sperm cells, which underwent mitosis to produce 2 haploid sperms, which produces a diploid zygote in the process and develops to become the embryo, while the other sperm moves up and fuses with the central polar nuclei, which forms a triploid cell and develops to become the endosperm, which serves as the embryo's food supply during embryo development. Meanwhile, the synergids degenerate after fertilization when it has done helping the release of sperm cells and cessation of the growth of a pollen tube, as well as the degeneration of antipodals after it has done nourishing the embryo sac and helps the development of endosperm. Overall, angiosperms only possess a double fertilization characteristic where a diploid zygote and triploid endosperm are formed. In conclusion, studying the plant reproductive system is fundamental for the fitness of the plant species, which means that a detailed knowledge can cultivate species and assess the adaptive significance and homology of descriptive characters used in plant systematics. Next is that the morphology of plants adapts and cooperates with the available pollinators in the environment, which means that factors coexist in order for the species to thrive that serves mutual symbiosis among species that entails succession of generations of both pollinators and fruit and seed production. Then the embryogenesis is a crucial phase for the plant life cycle, which tells us about the development of an effective management on the germination of the plants. And lastly, the plant development is essential for understanding the maturation and origin of plant species that would cultivate economy and find solutions in environments with barren diversity and struggling ecosystem.
It is recommended that in conducting the exercise, microscope and hand lens should be used in determining the microscopic structures of the floral morphology. Also, make a herbarium to preserve the collected plants for future use.